A few species such as Goniolax monolata form chains of similar cells with no differentiation. Their flagella have a strikingly characteristic arrangement in dinoflagellates. One long flagellum lies in a longitudinal groove with its distal end free and responsible for swimming. The other flagellum is flat and ribbon-like and lies completely within a transverse groove that encircles the cell. They differ from heteroconts by not having a tinsel flagellum. Most species are photosynthetic, but many are completely heterotrophic. Dinoflagellates have chlorophylls A and C, not A and B, and their carotenoids are quite unusual. In addition to beta-carotene, they also have several unique xanthophylls such as peridenin and dinoxanthin. Their reserve material is starch or oil. Dinoflagellates may lack a wall or have one consisting of cellulose plates whose arrangement and number are useful for identifying genera and species. During dinoflagellate sexual reproduction, vegetative cells release small, naked cells that act as gametes. At the initiation of fusion, they become quiescent but then resume swimming, and the zygote may remain motile for 12 to 13 days. It then becomes thick-walled and quiescent for approximately 2 months. During germination, it undergoes meiosis and a single haploid vegetative cell emerges, a new individual that grows and multiplies by mitosis. Organisms with many relictual characters are often regarded as poorly adapted, perhaps surviving only in certain specialized, protected environments, but dinoflagellates are abundant throughout the oceans. Under conditions of ideal temperature and nutrients, population growth of some dinoflagellates, especially Goniolax and Gymnodinium, is explosive. Within a few days they become so numerous that their bodies actually color the water reddish-brown, red tide. The density of dinoflagellates can be as high as 30,000 cells per milliliter of seawater. Red tides are becoming increasingly frequent in the Gulf of Mexico, a phenomenon that is unexplained at present. This fascinating biological spectacle is also dangerous because these algae produce toxins, and their large concentrations kill fish and make other marine life poisonous to humans. The poisons of many species, Goniolax catenella, for instance, are potent neurotoxins, interfering with the movement of sodium ions across our nerve membranes. Oomycetes. These protistans were thought to be fungi for many years. They have no chloroplasts, and thus, like fungi, they are never photosynthetic. Also, many of them have bodies that consist of long, slender multinucleate, cenocytic tubes that have no cross walls, an organization typical of fungi. However, many of their characters are more like those of algae and plants but different from true fungi. For example, Oomycetes have a cellulose wall but all true fungus cell walls are based on a polymer called chitin. Also, Oomycetes have tinsel flagella, and their chromosomes are fairly large. DNA sequences now strongly support inclusion of these groups in the heterocont protists. Oomycetes are diverse in structure and nutrition. Lacking chloroplasts, they must be parasitic or saprophytic. Most, such as Saprolegnia and Aclea, are aquatic and have been called water molds, but others live in dry habitats such as on and within leaves. Some consist of single cells that live as parasites inside rotifers, minute aquatic animals. Many Oomycetes, however, form extensive networks of slender tubular cells. Sexual reproduction occurs in most Oomycetes. A cell swells into a bulbous oogonium, which contains one or several diploid nuclei that undergo meiosis, each in its own mass of protoplasm. An adjacent cell elongates as an antheridium and contacts the oogonium, a hole forms, and microgametes pass directly into the oogonium, fertilizing the megagametes. The resulting oospores are diploid and are actually zygotes, each matures into a resting spore with a thick cellulose wall. These remain viable for many months even under dry, adverse conditions. Under favorable conditions, they germinate into diploid tubular cells. Figure 1935. Saprolegnia, an antheridium, has already contacted the oogonium and transferred microgametes. Oospores have formed. This situation is a form of oogamy. 2. Oomycetes are severe plant pathogens. The first attacks grapes and was introduced to France in the late 1870s on grafting stock imported from the United States. Plasmopara viticola caused tremendous damage throughout vineyards, except along roadsides where a mixture of lime and copper sulfate had been applied to the plants to keep passers-by from stealing grapes. After it was recognized that this inhibits the pathogen without damaging the grape plants, it was used for treating entire vineyards. Phytophthora infestans, an oomycete disease of potatoes, caused the Great Potato Blight and Famine in Ireland in 1846-1847. At that time, Ireland was overpopulated, and the potato was almost the only food for most people. The infection with P. infestans spread so rapidly and virulently that virtually the entire food crop for the country was destroyed. As many as 800,000 people starved to death the following winter, and more than 1 million emigrated, mostly to the United States. Phytophthora infestans is still a serious disease, and in cool, moist climates, it is controlled only by regular spraying with algicides. Hypochytridiomycetes are a related group of non-photosynthetic protistans that were formerly thought to be fungi. Euglenoids. More than 800 species of euglenoids have been discovered, named, and placed into 36 genera. The majority never have chloroplasts at all, but 11 do, derived through a secondary endosymbiosis with a green alga. Most euglenoids are unicellular, but in some species, a few cells remain together after cell division. Figure 1936. Euglena, the pellicle, now often called a periplast, is not a cell wall, but rather a thick layer of elastic proteins that accumulate just below the plasma membrane. Green, photosynthetic euglenoids have chlorophylls A and B, but carbohydrate is stored as the glucose polymer paramylon, not as starch. Euglenoid chloroplasts are extremely sensitive to antibiotics that interfere with 70S ribosomes, and exposure to streptomycin results in the loss of chloroplasts. Colorless, bleached individuals can be produced with this treatment. Many colorless individuals, both natural and bleached, take in food particles by phagocytosis, and most absorb sugars and even proteins through their plasma membrane. Even chlorophyllous species grow faster in light if supplied with organic compounds, and all must obtain B vitamins from the environment. Euglenoids swim actively with two flagella located at the cell's anterior end, one short and the other quite long. Euglenoids differ from other protists and plants in an important character: they never have a cellulose wall. Instead, their surface contains a layer of elastic proteins that constitute a pellicle, which is located under the plasma membrane and gives the cell some rigidity and shape. Euglena is a representative example. Individuals are elongate cells that, because of their pellicle, have a somewhat ovoid shape, although they flex as they swim. At the anterior end is an invagination, the canal. The two flagella are attached at the base of the canal and extend upward, but only one is long enough to emerge from the canal and be useful for swimming. At the side of the canal, near the base of the flagella, is an eye spot composed of many small, orange droplets of carotenoids. There are numerous disc-shaped chloroplasts, each containing a region called a pyrenoid. In most algae, the pyrenoid is involved in polymerizing sugars into reserve polymers. That may also be true in Euglena, but the reserve paramylon accumulates outside the chloroplast, away from the pyrenoid. Euglenoids have many features that must have undergone little change and thus probably are similar to those present in early eukaryotes. During mitosis, their nuclear envelope and nucleolus persist, and spindle microtubules form within the nucleus. Chromosomes are aligned lengthwise on the spindle, not crosswise as in other eukaryotes, and each chromatid appears to have its own mini-spindle consisting of 4 centromere microtubules and 8 to 12 longer microtubules. Cell division is by longitudinal cleavage, not by either a phragmoplast or a phycoplast, and after division is completed, chromosomes remain condensed. Neither meiosis nor sexual reproduction has been found in any euglenoid. Many features of normal mitosis and meiosis as occur in plants and animals apparently did not arise until after the euglonoid clade had become reproductively isolated from the rest of the early eukaryotes. At the next level. 1. Complex multicellular bodies in algae. Both red algae and brown algae have complex bodies composed of several distinct tissues. Each evolved separately, so each presents many new alternatives for constructing a complex sessile body. Both have complex meristematic and reproductive tissues, and many brown algae conduct sugars through tissues that resemble phloem but which originated independently. It is the result of convergent evolution with phloem. Studying these complex algal bodies will give you a more complete appreciation for the bodies of true plants. 2. Red tides. In many cases, organisms become more complex as they evolve to become more adapted. But this is not always true. Many of the unicellular algae are so well adapted to ocean habitats that nothing outcompetes them, and when conditions are just right, unicellular algae become so abundant they cause red tides. Some of the red tide dinoflagellates are poisonous and kill animals in the area, altering the community biology. As we put more sewage and fertilizers into rivers that empty into oceans, and as global temperatures rise, red tides are becoming more frequent and may have a permanent effect on coastal ecology. 3. Acetabularia. Complex giant cells with a single nucleus. Many algae have unicellular bodies, but species of acetabularia are spectacular. Each cell is gigantic, almost 1 centimeter long, and often have a stalk, a cap, or finger-like extensions, and despite the complexity of such cells, they have just one nucleus. Many ingenious experiments were performed in the early days of developmental biology in which nuclei were transplanted from a cell with one shape into the enucleated body of a cell that had a different shape. After a short recovery, the cells would change their shape to correspond to the instructions from the new nucleus. At present, we take such things for granted, but at that time this was extraordinary proof of the importance of nuclei. Reading about the research performed with acetabularia will expand your appreciation of the ingenuity and talent needed to discover the facts we rely on today. Summary. 1. Several significant evolutionary divergences occurred in the early eukaryotes, long before the plant, animal, and fungus clades had originated. Euglonoids have no meiosis or sexual reproduction. Dinoflagellates lack histones and normal mitosis. Several groups have surfaces that lack cellulose or have cellulose combined with chemicals not found in plants. 2. A primary endosymbiosis produced the chloroplasts of glaucophytes, red algae, green algae, and plants. Several events of secondary endosymbiosis of entire red or green algae produced chloroplasts of the other protists. 3. Algae are distinguished from plants by having gametangia and sporangia in which fertile cells are not protected by a surrounding layer of sterile cells. 4. Cell division in unicellular algae is by membrane infurrowing and cell cleavage. In some green algae, it is by means of a phycoplast. In most multicellular algae, cell division occurs by phragmoplast and outward growth of the cell plate, but in red algae, walls grow inward from the existing side walls. 5. Charophytes have a flagellar root complex of one large band of microtubules, similar to that in plants. Other green algae have a root complex of 4 bands of microtubules, unlike that of plants. 6. Within green algae, several body types have evolved from unicellular forms, modal and non-modal colonies, branched and unbranched filaments, coenocytes, membranous forms, and true parenchyma. 7. Several types of life cycle exist: dibiontic, in which both a diploid and a haploid generation occurs, monobiontic, in which only one generation is capable of mitosis. 8. Dibiontic life cycles may consist of an alternation of either isomorphic or heteromorphic generations. All plants have an alternation of heteromorphic generations. 9. Several evolutionary clades are evident in the green algae, but the charophyte clade is the one that most strongly resembles embryophytes. 10. Brown algae and their relatives make up the heterocont clade, sharing the synapomorphy of having one tinsel flagellum and one whiplash flagellum. Important terms. Agar. Alternation of heteromorphic generations, alternation of isomorphic generations, anisogamy, archaeplastid. Autogenous theory, brown algae, charophytes, coccolithophorids, cenocytic body colony conceptacles, conjugation, diatoms, dibiontic dinoflagellates, embryophytes, endosymbiont theory, euglenoids, filamentous body, floridian starch, gammatangia, golden brown algae, green algae, heteroconts, holdfast, isogamous, laminar and literal. Zone membranous body, monobiontic species, oogamy, oomycetes, parenchymatous body, phycoplast, pit connections, nematocyst, primoplanty receptacles, red algae, red tide, sporangia, streptophytes. Trumpet cells, yellow-green algae, zoospores. Review questions. 1. All organisms are classified into 3 domains. Name the 3 and describe the organisms that are classified in each. 2. Reproductive structures are the critical factors in distinguishing algae from true plants of kingdom Plantae. What is one fundamental difference between reproductive structures in these 2 groups? 3. Which group of algae appears to be most closely related to the ancestors of true plants? Which features appear to be homologous? 4. Describe the endosymbiont theory of plastid origin. How many times might chloroplasts have arisen? 5. In contrast to the endosymbiont theory is the autogenous theory. Describe the basic idea of it. Do you think that the autogenous theory would predict that plastids and mitochondria would have DNA and 70S ribosomes? 6. Why do we think that cyanobacteria rather than purple or green bacteria gave rise to chloroplasts in the primary endosymbiosis? Which cyanobacteria are most similar to chloroplasts? Which algae were involved in the secondary symbioses? 7. Question 6 indicated that chloroplasts arose several distinct times in separate acts of endosymbiosis. If that is true, does the term plastid refer to a natural group of organelles that descended from a common ancestor? Would it be safer to use 3 separate names for these 3 separate structures? 8. List some of the unusual cyanobacterial features of red algal chloroplasts. 9. Describe cell division and mitosis in algae. How is the red alga method of cross-wall formation different from that of plants? 10. If after cell division the cells adhere to each other loosely, the resulting structure is not an individual organism. In such a structure, are there many types of different cells or are all cells more or less similar to each other? 11. Name and describe the 6 types of body construction that occur in green algae. What role does the middle lamella play in these? What is the importance of controlling the orientation of cell division? Which of these body types occurs in other groups of algae? 12. Describe monobiontic and dibiontic life cycles. Be careful to mention all possible types. What is the difference between a spore and a gamete? What is the difference between a spore and a zygote? 13. What is an alternation of isomorphic generations? What is an alternation of heteromorphic generations? If you have studied the life cycle of flowering plants, do flowering plants have an alternation of isomorphic or heteromorphic generations? 14. Describe a green alga with a life cycle involving alternation of heteromorphic generations. Why were the 2 generations originally named as separate species? 15. Volvox is a green alga that you might see in a biology or botany lab. Is it filamentous or membranous or colonial? What is its shape? 16. Spirogyra is another green alga you might see in a biology or botany lab. What is unusual about it? What gives it its name? 17. Chara is an unusual alga. What type of cell division do members of Charophyceae undergo, division with a phycoplast or phragmoplast? What is important about their flagella? Do true plants have flagella? 18. Red algae are unusual in many ways, but an especially significant trait is the types of accessory pigments in their chloroplasts. What pigments do they have? They are aggregated into a body called a. 19. Many red algae have cell walls so heavily impregnated with calcium carbonate that they are brittle. These are called the red algae. 20. What are trumpet cells in brown algae? What type of cells do they resemble in true plants? Is their presence in brown algae a sign that brown algae are related to the ancestors of true plants, or is it just a remarkable case of convergent evolution? 21. If you have ever visited a rocky coast, you may have seen two brown algae, kelps and fucus. Briefly describe the bodies of these two common seaweeds. 22. Dinoflagellates are abundant algae with many unusual features. The arrangement of their flagella is characteristic. Describe the flagella of a typical dinoflagellate. 23. Under ideal conditions, dinoflagellates can reproduce so rapidly that they actually give seawater a distinct color. What is this phenomenon called? Is it safe for fish and humans? 24. Diatoms have what chemical in their cell walls? When the cell dies and the protoplasm degenerates, the wall sinks to the ocean bottom and becomes part of a huge deposit known as. These deposits can be so extensive because diatoms are so abundant that just a single liter of sea water may easily contain more than individuals. 25. Describe and compare the cell walls or cell coverings of dinoflagellates, euglenoids, diatoms, green algae, red algae, and true plants. 26. Describe features of oomycetes that caused people to believe they were fungi. Now describe feature that indicate they are heterocont algae. Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Chapter 20. Non-vascular plants, mosses, liverworts, and hornworts. Chapter opener image: is this a plant or an alga? It certainly looks like a green alga at first glance, but one hint that it is a plant is that it has many small chloroplasts per cell, whereas most algae have only 1 or 2 chloroplasts per cell. When spores of mosses, liverworts, and hornworts germinate, the first cells that grow out strongly resemble filamentous green algae. This was one of the characters that led early botanists to suspect that green algae are the closest relatives of true plants. After this moss grows slightly larger, some of the tip cells will convert themselves into apical cells in the shape of an inverted pyramid. After this, all growth will be obviously plant-like with stems and leaves. Outline. Concepts. Characters of non-vascular plants. Classification of non-vascular plants. Division: Bryophyta, mosses. The gametophyte generation. The sporophyte generation. Metabolism and ecology. Division: Hepatophyta, liverworts. The gametophyte generation. The sporophyte generation. Division: Anthoceratophyta. Hornworts. The gametophyte generation. The sporophyte generation. Learning objectives. After reading this chapter, students will be able to summarize the changes and adaptations that allowed for the evolution of non-vascular plants. List 4 characteristics of non-vascular plants. Explain the morphology, water transport, development, and reproduction of mosses. Recall how water availability and retention affect moss sustainability. Describe liverwort gametophores. Compare liverwort sporophytes to moss sporophytes. Recall 4 characteristics of hornworts. Note how hornwort sporophytes differ from liverwort and moss sporophytes. Did you know? Many species of mosses and liverworts, when viewed with a hand lens, are as easy to recognize as wildflowers. The green moss plants you see are haploid, the small tan or brown capsules and stalks they sometimes have are diploid. Many mosses are adapted to deserts. Dew forms on their tips on cool mornings and is conducted downward along the exterior of the moss. Many of the characters of hornworts and some liverworts are very similar to those of the first true plants. Concepts. In this chapter, the origin and evolutionary diversification of true plants, embryophytes, are discussed. Several critical events occurred in the history of true plants: their origin from green algae, their adaptation to terrestrial habitats, the development of the vascular tissues xylem and phloem, and the origin of seeds, leaves, and woody growth. Plants are traditionally divided into those that have neither vascular tissues nor seeds. The non-vascular plants have vascular tissue but not seeds, the vascular cryptogams, and have both vascular tissue and seeds, spermatophytes. Non-vascular plants arose first, and some of their later members were the ancestors of the vascular plants. Many of the fundamental aspects of plant biology were established in the early land plants. Beginning at some point earlier than 450 million years ago, certain green algae known as charophytes began to adapt to living on land. They evolved to survive the occasional drying of their streams, smaller lakes, and oceanside mud flats. In environments where long, dry periods alternated with long, moist periods, the optimal survival strategy probably was formation of dormant, drought-resistant spores, but spore production requires major metabolic conversions, if the dry periods were brief. The ability to continue active metabolism by conserving water and avoiding desiccation would have been advantageous. A large, compact, multicellular body would have had a low surface-to-volume ratio and would have automatically retained water better than a small unicellular or filamentous body. In addition, a waterproofing cuticle would have been selectively advantageous. Reproduction would have had to change to coordinate gamete production with periods of moisture because sperms had to swim. Also, gamete and spore mother cells would have needed protection from dryness by having a jacket of one or several layers of cells around them. Such reproductive modifications resulted in the grouping and protection of spore and gamete mother cells into the sporangia and gametangia characteristic of all embryophytes, more massive than those of algae and protected by a layer of sterile cells. Table 21 Major groups of plants. Figure 21 Recently proposed phylogeny of true plants. Table 22 Means of coping with dry periods. These simple modifications probably allowed the algal ancestors of plants not only to survive but also to be metabolically active during short dry periods. However, an automatic consequence of much greater advantage was that the algae were safe from predators while out of the water. Tremendous selective benefit resulted from mutations that enabled the algae to be active for even longer periods out of water. These mutations probably involved increasing the size of the body and the impermeability of the cuticle, but there had to be the simultaneous evolution of stomatal pores and guard cells because a more protective cuticle also prevents the entry of carbon dioxide. Figure 22 In true plants, gametangia and sporangia always have an outermost layer of cells that do not become gametes or spores. In these microgametangia of mosses, the sterile layer is only one cell thick, and each microgametangium produces numerous sperm cells. Compare this with an algal gametangium. As a truly terrestrial existence became more successful, the mud flats and stream banks would have become crowded, and some plants must have grown over others and shaded them. With such shading, the environment became selective for mutations that produced an upright body that could grow into brighter light. The non-vascular plants mostly use turgid parenchyma cells and thus cannot grow very tall, but in the group of plants that would become vascular plants, the strengthening material that evolved was xylem, which is also good at conducting water. As phloem evolved, the basal part of the plant that remained in the shade could be nourished. Phloem permitted roots to extend deep into the dark soil, and xylem permitted transport upward of the water and nutrients those roots encountered. The presence of xylem, phloem, and roots had profound consequences. They freed plants from their muddy habitats and allowed them to grow anywhere moisture was available. The presence of the two vascular tissues, necessary for individuals even a few centimeters tall, suddenly gave plants the potential to become 100 meters tall, to branch, to form leaves, and to put sporangia high into the air, allowing the spores to be blown great distances, carrying the plant's genes over huge areas, and permitting the colonization of distant habitats. Figure 23. Megagammetangia of true plants, such as these of a moss, also have a one-layered sterile jacket, but each produces only one egg, not four. This scanning electron micrograph shows the gammetangia of a bryophyte. The spheres are microgammetangia in which sperm cells are produced, and the tubular structures are megagammetangia, each with a single egg inside. The large flat sheets of cells are the leaves of this leafy liverwort. Vascular tissue, especially phloem, also made feasible the evolution of truly heterotrophic tissues, roots, meristems, and organ primordia. Without phloem, each part of a plant can grow and develop only as rapidly as its photosynthesis permits, but phloem allows mobilization of sugars, minerals, and hormones throughout the entire body in their transfer to a shoot apical meristem or a group of sporangia, thus permitting a more vigorous, robust growth than could otherwise occur. One obstacle to the total invasion of land was that plants were still reproductively amphibious. They had terrestrial bodies but aquatic reproduction with swimming sperms. At some point in the life cycle, the environment had to be sufficiently wet that sperms could swim from one gametophyte to another. This requirement is a handicap, but not an insurmountable one. All plants need water to grow. No plant ever lives where there is never rain, fog, or dew. Production of pollen and seeds eliminates the need for environmental water for reproduction. What changes must occur for seeds to evolve? Gametophytes must become so reduced that they mature completely within the walls of the spores. After this happened, retention of the megaspore and megagametophyte inside the parental sporophyte was feasible. The gametophytes functioned basically as tissues of the sporophyte and benefited from the land adaptations that had evolved. This is what occurs in ovules of seed plants. The microgametophyte, inside the microspore, could be transferred by wind to the vicinity of the megasporangium, where it released sperms into fluids secreted by the megagametophyte. The sperms would swim in this tiny artificial pond and effect fertilization. Only the last step of reproduction was still wholly aquatic. In the most derived seed plants, a pollen tube carries non-motile sperms even further, to the egg itself. Some gametophytes, for example, those of all non-vascular plants, do not need any protection by the parental sporophytes, in mosses, liverworts, and hornworts. Gametophytes are the main photosynthetic phase of the life cycle, but even so, these gametophytes remain rather small and delicate, limiting the entire plant to environments mild enough for themselves. Plants should have been able to develop a life cycle in which gametophytes were just as large, tough, and woody as sporophytes, even having bark, leaves, roots, and so on, but this never happened. Some early land plants did have rather large gametophytes, but in all lines of evolution, gametophytes became simpler and smaller. Figure 24. This rock is covered with a moss and lichens. Mosses typically grow in moist, shady habitats. However, there are also large numbers adapted to dry, sunny conditions as here at Inks Lake State Park in central Texas. This moss thrives side by side with cacti and yuccas in the blazing sun and with only sporadic, infrequent rains. But the important thing is that it does rain at some point, and there is dew or fog at other times. That is true of absolutely every place where plants grow. No plant ever grows where there is never liquid water at some time. Mosses always have small, simple bodies, and rather than being a handicap, this is an adaptation that allows them to live in small, rocky areas that have only a bit of occasional water, too little water to sustain a large tree. Mosses are confined to moist areas because their sperm cells must swim from one plant to another. The sperms do swim, but there is always liquid water at some time, so sexual reproduction is possible. It is important to learn the characteristic features and habitats of plants, but it is also important to be aware of alternative adaptations and their consequences. Figure 25. The megagametophyte of Selaginella, which has developed almost completely within the original wall of the megaspore. This is not part of a seed plant but is a necessary first step in seed evolution. Small megagametophytes can be protected and nurtured by their parent sporophyte. By doing so, the gametophyte generation benefits from all of those mutations that make the sporophyte adapted for life on land. Characteristics of non-vascular plants. Most people are at least somewhat familiar with non-vascular plants because mosses are well known, and many people have heard of liverworts. The third group, hornworts, is relatively unfamiliar, even to most botanists. It is important to understand clearly what non-vascular plants are not. Spanish moss of the southeastern United States is the flowering plant Tillandsia usneoides of the pineapple family, not a true moss. Club mosses are lycophytes, not mosses, and the slimy, bright green mosses of ponds and slow-moving streams are green algae, usually Spirogyra. Several types of lichens, especially Alectoria, Bryonia, Usnea, and reindeer moss, are frequently mistakenly thought to be mosses or liverworts. Figure 26. The tiny leafy shoots are gametophytes of a small moss. The numerous plate-like structures are gametophytes of a hornwort, Phaeoceras. Plants of both remain small and thin, never more than a few cells thick, and never elaborate. Figure 27. Although mosses occur in hot, arid areas, they are most abundant in cool, moist regions. What you think of as the typical moss plant, green or gray-green and leafy, are masses of gametophytes. The slender stalk and capsules you may have noticed are sporophytes. Technically, non-vascular plants are embryophytes that do not have vascular tissue. Being embryophytes, they have multicellular sporangia and gametangia. Reproductive cells are always surrounded by one or several layers of sterile cells. Their bodies are not composed of filaments as in many algae, but rather of true parenchyma derived by three-dimensional growth, usually from apical meristems. All mosses and many liverworts have leafy stems that look remarkably like small versions of flowering plants. Non-vascular plants are almost exclusively terrestrial and have a cuticle over much of their bodies, and many have stomata. Figure 28. This is Marchantia, the most abundant greenhouse liverwort. Unfortunately, it is one of the least typical. As in mosses, the large green plant is the gametophyte, not the sporophyte. The cup-shaped structures contain clumps of cells that can be splashed out by rain and then grow into new plants. Figure 29. Hornworts such as this Phaeoceras are quite rare, and few people ever see them. They are easily confused with liverworts unless the tall horns, sporophytes, are present. A assumes that they are less closely related than B does. Like all plants, non-vascular plants have a life cycle with an alternation of heteromorphic generations. The sporophyte and gametophyte differ from each other structurally. Recall that sporophytes in flowering plants are the large plants with leaves and roots, whereas gametophytes are tiny and occur inside pollen grains and ovules. In non-vascular plants, the gametophyte is the larger, more prominent generation, and the sporophyte is much smaller, more temporary, and often very inconspicuous. Any green moss plant that might be familiar to you is the gametophyte, the haploid phase, and it is photosynthetic, perennial, and collects mineral nutrients. It typically can spread rapidly by asexual reproduction. The sporophytes it produces are usually very small. They absorb minerals only from the gametophytes and always remain attached to them. Sporophytes carry out so little photosynthesis that they could not support their own growth and sporogenesis. Unique sporophyte features in all species of non-vascular plants are that sporophytes are never independent of the gametophyte, never branch, and never have leaves. You will see sporophytes only if you examine mosses closely. they look like green or brown hairs standing up on the green gametophyte, but sporophytes are only present at certain times of the year. Non-vascular plants can never grow to be really large, but being small and simple provides great selective advantage in certain habitats. The tiny parenchymatous bodies of mosses and liverworts permit them to thrive in microhabitats such as stone walls, fences, and bare rock, microhabitats that have too little water or soil for the larger, more complex vascular plants. Table 23 Alternative methods of classifying non-vascular plants. Classification of non-vascular plants. It is not known how closely related mosses, liverworts, and hornworts are. They have many features in common but also differ in significant respects. They are often treated as three distinct divisions: liverworts, division Hepatophyta, mosses, division Bryophyta, and hornworts, division Anthoceratophyta. In the past, all three were often grouped together in division Bryophyta as three classes: mosses, Mussae, liverworts, Hepaticae, and hornworts, Anthoceroti. Even today, they are informally referred to as bryophytes. The three are treated here as divisions, and the generic term bryophyte is used only for mosses. All groups together are called non-vascular plants. Division Bryophyta, mosses. The gametophyte generation. Mosses are ubiquitous, occurring in all parts of the world and in almost every environment. They are perennial and thrive in many places within cities. Morphology. The leafy stems, technically known as gametophores, of many moss plants grow close together, tightly oppressed and forming dense mounds. In other species, particularly those of cool wet areas, the plants are more open and loose. Scleria gametophores grow as ribbons up to 15 centimeters long, submerged in rapidly flowing water. All moss stems have leaves, but because they are parts of a gametophyte, not a sporophyte, they are not homologous with those of vascular plants. That is, leaves of mosses did not evolve from the same structures as vascular plant stems and leaves did. Table 24 Classification of division Bryophyta. Moss gametophores grow from an apical meristem that contains a prominent apical cell. Derivative cells subdivide, producing the tissues of stem and leaves in rather precise arrangements. Leaves are aligned in 3 rows at least while young, and most have a midrib. Leaves of almost all mosses are only 1 cell thick except at the midrib and along the margin. In the family Polytrichaceae, common along roadsides in forested areas of the northern United States, genera such as Polytrichum and Atrichum have thin lamellae on the leaf upper surface, greatly increasing the photosynthetic tissue. Cuticle occurs only on the upper surface of most moss leaves, the underside being uncutinized and capable of absorbing water directly from rain, dew, and fog. The lack of a cuticle means that mosses have little protection against desiccation. When the microhabitat dries, so do the leaves. No stomata occur on moss leaves, they would be useless because the leaves are unistratose. Figure 2010 The individual shoots of a moss superficially resemble those of a flowering plant, having stems, leaves, nodes and internodes, and even buds. None of these structures is homologous to the equivalent organ in flowering plants, but the same set of names is used for both groups. Dark green, haploid tissue, light green, diploid tissue. Moss stems are always slender and have little tissue differentiation. The surface layer is at most only slightly different from the underlying layers and is not called epidermis. The stem tissues, all called cortex, may be uniform in all parts, or the outer cells may be slightly narrower with walls that are somewhat thickened. Inner cells are larger, more parenchymatous, and chlorophyllous. In a few species of mosses, stems have hairs, but stomata do not occur. Figure 2011 in cross-section of the shoot apex of the moss Physcomitrium pyriforme, the apical cell is in the center, and its most recent daughter cell is to the left. The next older cell has subdivided, as have successively older derivatives. The apical cell is shaped like an inverted pyramid. Figure 2012. Many moss leaves are only one cell thick, but the leaf midrib is thicker and is true parenchyma. In the family Polytrichaceae, leaves bear long sheets of cells on their upper surface. This greatly increases the volume of photosynthetic tissue but does not decrease the high surface-to-volume ratio as it would if this were a solid tissue. Water transport. In some mosses, primarily the family Polytrichaceae, the innermost cortex is composed of cells called hydroids that conduct water and dissolved minerals. They are elongated cells that lose their cytoplasm when mature. Their end walls are partially digested, but they are not removed entirely. Each hydroid is aligned with those above and below it. Species that have hydroids typically also have leptoids, cells that resemble sieve cells. They are elongate, have relatively prominent interconnections with adjacent cells, and lack nuclei at maturity, although they do retain their cytoplasm. Adjacent parenchyma cells are unusually cytoplasmic and rich in enzymes, just as are companion cells. The majority of mosses lack hydroids and leptoids. Water is conducted along the exterior of their stems by capillary action. Leaves and stems are so small they form spaces narrow enough to act as capillary channels and transport water. In some species, such as Funaria, leaves curl and become more closely oppressed to the stem as they dry. When rain or dew occurs, the dried plant has even more capillary spaces than in the hydrated condition when the leaves are spread away from the stem. In mosses that lack leptoids, sugar is moved simply between parenchyma cells by slow transport. Figure 2013. Although most moss stems do not have vascular tissues, they do support the shoot, and most have a layer or two of thick-walled cells. Sugars and minerals must be transported from leaves to the shoot apex, gametangia, and sporophytes. Therefore, living parenchyma cells are necessary. At the base of the stem are rhizoids, small, multicellular trichome-like structures that penetrate the surface of the substrate. Rhizoids only anchor the stem. They do not absorb either water or minerals. They lack chloroplasts and have reddish walls. Figure 2014. Hydroids lose their cytoplasm at maturity, just as do tracheids and vessel elements. Hydroids, however, do not have specialized secondary walls. Leptoids are analogous to sieve cells, phloem cells that are simpler than sieve tube members. All pores are small, and the cells remain alive at maturity. Development. Growth of the gametophore begins when a spore germinates and sends out a long, slender chlorophyll cell. This cell undergoes mitosis and produces a branched system of similar cells. The entire network is a protonema. A protonema superficially resembles a filamentous green alga but can be distinguished by its numerous small chloroplasts in each cell. Algal cells have only one or two large chloroplasts. Eventually, nodules of small cytoplasmic cells form on the protonema, organize an apical cell, and then grow upright as a stem with leaves, the gametophore. Protonemata are perennial and can grow extensively, producing many buds. The filamentous cells usually break when mosses are collected, and thus, a tuft of gametophores may appear to be independent plants when in fact they have all arisen from a single protonema. Reproduction. The gametophore at some point produces gametangia. All mosses are oogamous, that is, every species has small biflagellate sperm cells and large non-motile egg cells. Sperms are produced in microgametangia called antheridia, which consist of a short stalk, an outermost layer of sterile cells, and an inner mass of cells that differentiate into sperm cells. Eggs occur in megagametangia called archegonia. Each is shaped like a vase with a long neck. The neck is hollow at maturity, and the single egg is located at the base. Antheridia and archegonia occur on the same gametophore in bisexual species, whereas other species have both male and female gametophores. Depending on the species, gametangia may occur mixed with leaves along the gametophore stem or may be clustered at the stem tip, surrounded by a special cup-shaped whorl of leaves. Raindrops fall directly into the cup, splashing sperms out and carrying them as far as 50 centimeters. Without this mechanism, sperms must swim to archegonia. Figure 2015. A moss spore has germinated and grown into these filaments of cells, which make up a protonema. Later some cells will divide into apical meristems that will grow into gametophores. Although this looks like a green alga, algae do not have so many chloroplasts in each cell. When sperm cells are mature, the antheridium breaks open and liberates sperm cells either by contracting the sterile outer cells or by accumulating liquid below the sperms and pushing them out. Secretion of sucrose from the archegonia guides sperms as they swim toward the archegonia and then down the neck to the egg, where one sperm cell effects fertilization. The sporophyte generation. In all embryophytes, the megagamete and subsequently the zygote are retained by the gametophyte. However, unlike those of mosses, megagametophytes of vascular plants are small and offer little nourishment or protection to the embryonic sporophytes. Megagametophytes of flowering plants typically have only 6 cells other than the egg, and both synergids and antipodals often degenerate just after the egg is fertilized. All nutrition for the zygote is supplied by the grandparent generation, the sporophyte, by way of endosperm formation. In contrast, moss gametophytes are both large and photosynthetic, and they support the sporophyte throughout its entire life. The moss sporophyte is never an independent, free-living plant. The zygote of a moss undergoes a transverse division, and the basal cell, located at the bottom of the archegonium, develops into a small, bulbous tissue called the foot. The foot is the interface with the gametophore, from which it absorbs sugars, minerals, and water. Its cells are transfer cells in many species. The upper cell grows by cell division and expansion into a simple apical sporangium called the capsule, consisting of an outer layer of sterile cells and an inner column of sterile cells. A ring of sporogenous cells undergoes meiosis, producing haploid spores. Between the foot and the sporangium is a narrow stalk, the seta. All moss sporophytes have this basic, simple structure. None is ever branched or has leaves, bracts, or buds of any kind. Although morphologically simple, the sporophyte is relatively complex structurally. It has a true epidermis with stomata at least on the base of the sporangium. Dehiscence of the sporangium is more elaborate than the opening of the gametangia. The apex of the sporangium differentiates as a cap-like lid, the operculum, which separates from the rest of the sporangium as cells are torn apart. Cell breakage is elaborate and precise, resulting in 1 or 2 rows of beautiful, exquisitely complex teeth, called peristome teeth. The teeth respond to humidity, bending outward and opening the sporangium when the air is dry and bending inward and trapping the spores when the air is humid. Spores are released when they are light, dry, non-sticky, and easily carried by air currents. The apex of the sporangium in many species is covered by a calyptra, a layer of cells derived from the neck of the archegonium. As the embryo begins to grow, neck cells also proliferate. They keep pace with sporophyte growth at first but later grow more slowly and are torn away from the gametophore. Figure 2016 Life cycle of a moss. Details are given in the text. Virtually all mosses are homosporous. All spores are the same size and appear to be identical. A few species of Macrometrium and Schlothemia produce 2 types of spores in each capsule. The larger spores develop into large gametophytes with archegonia, and the smaller spores grow into dwarf males that live epiphytically on the females. Metabolism and ecology. The small size and lack of conducting tissues are 2 critical factors in the metabolism and ecology of mosses. Vascular plants tend to be large and their bulk protects them from short-term fluctuations in air humidity and moisture availability. Should the air become dry for several hours, most seed plant leaves and stems would not die, even if they were not receiving water from the roots by means of xylem. Very large plants, such as trees and succulents, can withstand dry conditions for weeks, months, or even years before losing a fatal amount of water. Leaves on moss gametophores, however, have only a thin, incomplete cuticle. If exposed to dry air for even a few minutes, the plants dry out. Without vascular tissue, stems and leaves can become desiccated, even while the rhizoids are in contact with moist soil or tree bark. Figure 2017. In this moss, antheridia are clustered together at the gametophore apex. In other species, they might be located along the stem. The phenomenon of dwarf males is common in many plants, animals, and algae. Each sperm cell is much smaller and less expensive to produce than each egg, but usually, many sperm cells are not carried to eggs, thus, a large number of sperm cells must be produced. If sperm delivery can be ensured, the number produced can be decreased, and even a small, dwarf male can produce adequate numbers. Delivery can be assured if the male gametophyte grows on the female as an epiphyte. Several mechanisms compensate for the inability of mosses to retain water. Many species grow in permanently moist microhabitats such as rain forests, cloud forests, and the spray zones near waterfalls. Even habitats that are not constantly humid may have microhabitats that are continually moist. The foot of a rock cliff is usually damp because dew and mist concentrate there as runoff from the cliff. Shallow depressions in rock or soil retain moisture and are protected from drying winds. These sites are very small, but so are the mosses that live within them. Other mosses are tolerant of desiccation. Drying does not damage them as it does most vascular plants and algae. Like lichens, many mosses can lose much of their water rather rapidly without dying or even being injured. As long as about 30% of their weight is water, they remain dormant but alive. If rain falls or dew forms, water is absorbed rapidly, and within a few minutes, respiration and photosynthesis are occurring at normal levels. In mild temperate regions, mosses may remain moist and metabolically active except for certain times during summer months. In deserts and dry rangelands, on the other hand, mosses may be turgid primarily during winter. From spring to autumn, the mosses are dry and inactive every day, perhaps receiving enough dew on certain nights to be metabolically active for a few hours in the early morning. Desiccated mosses are remarkably resistant to high or low temperature and to intense ultraviolet light. A common moss, Tortula ruralis, can be frozen safely in liquid nitrogen when dry and can also withstand brief periods at plus 100 degrees Celsius. Resistance to UV light is important for species that grow in full sun on the surface of rocks or at high altitudes on mountains, where the atmosphere is too thin to block UV radiation. Figure 2018. The organelles of a young sperm cell have rather typical shapes. During differentiation, the organelles become modified, resulting in an elaborate spiral-shaped sperm cell at maturity. Cytosol and cell membrane are not shown. Figure 2019. Aspects of a moss sporophyte. The foot is embedded in the remnants of the archegonium. External view of the capsule with the operculum still in place covering the teeth. Median section view showing the columella and spores. External view after the operculum has fallen off. Spores can be shed when the peristome teeth bend back. Scanning electron micrograph view of peristome teeth. Many moss species thrive at low temperatures near or even below 0 degrees Celsius. When larger vascular plants die or abscise their leaves in winter, much more sunlight reaches the soil surface or tree trunks where mosses are growing. Snow or winter rains provide abundant moisture. Mosses that can carry out metabolism at cold winter temperatures can take advantage of the plentiful light and moisture. Certain mosses can grow on hard, impervious surfaces because they have no roots that must penetrate the substrate. Consequently, mosses can be found on bare rock, brick or mortar, and bark as well as soil. At the other extreme, sphagnum mosses live on the surface of quiet water at the edges of lakes and ponds. Their leaves and stems are buoyant, and thick mats build up. In almost all environments, mosses are important in the later establishment of other species, particularly vascular plants. Like lichens, they can colonize a bare surface and dissolve the rock with acids that leak from their cells. Leaves and stems of gametophores catch and hold dust particles, creating small pockets of soil in which spores and seeds of vascular plants can become established. A similar process occurs even on the ground. In forests or grasslands, the moss layer on the soil may be thick enough to act as a moist, airy seed bed. It may hold water better than the soil and thus improve the microhabitat for seedlings. Figure 2020. Sphagnum moss grows at the edges of streams or ponds where there is little water movement. The plants become large and tangled, creating a mat that is solid enough to support the weight of an animal. These are known as quaking bogs because they quake and undulate with every step. Given enough time, seeds of vascular plants germinate, and leaves and debris blow onto the bog and decompose, slowly converting it to a rich soil. Division Hepatophyta, liverworts. Like mosses, liverworts are small plants that have an alternation of heteromorphic generations. The smallest individuals of Cephaloziella are only 150 by 2000 mm. Few species have plants that ever become large, the maximum being approximately 5 by 20 cm in monoclea. Some species are leafy and greatly resemble mosses, others form small, solid, ribbon-like gametophytes. The sporophyte is even less conspicuous than in mosses and is also completely dependent on the gametophyte. The gametophyte generation. Hepatic gametophytes are divided into two basic groups, leafy liverworts and thallose liverworts. In both, the gametophyte phase is initiated when spores germinate and establish a small, temporary protonematal phase. Liverwort protonemata are never as extensive or ramified or as long-lived as those of mosses. Instead, after only a few cells are produced, an apical cell is established and growth of the gametophore begins. The gametophore of leafy liverworts greatly resembles that of a moss, thin leaves on a slender stem. However, liverwort leaves typically have two rounded lobes with no midrib and no conducting tissue, whereas moss leaves are pointed and usually have a midrib. Liverwort leaves are arranged in three clearly defined rows, the leaves of two rows being much larger than those of the third. In prostrate liverworts, in which the shoot grows flat against the substrate, the underside row of leaves is reduced and may even be completely suppressed or replaced by rhizoids. Table 25. Classification of division Hepatophyta. Figure 2021. Leafy liverworts such as this Lophicolia can be very easily confused with mosses. Their distinguishing features are their typically lobed leaves, which grow from two apical points, and their basal pouch. Moss leaves are never like this. Thallose liverworts have rather thick bodies and are not easily confused with mosses. This is a moist, active oxymetra incrustata, notice its papery scales. White dots are air pores, clefts contain gametangia. When young, the gametophyte had one apical meristem and one cleft, and then the apex divided dichotomously, so two apices and two clefts are now present. Each can divide again. In dry seasons, plants of oxymetra desiccate and fold inward, bringing the scales over their body, protecting themselves. They survive months of severe heat and drought. Figure 2022. Life cycle of Marchantia. Details are given in text. The gametophore stem grows by an apical cell with 3, 4, or 5 sides, one uppermost and the others embedded in the stem. Stem tissue is just simple parenchyma. No thick-walled cells or conducting cells occur in the majority of species. Virtually all liverworts have some or many cells that contain characteristic oil bodies. Thallose liverworts show less resemblance to mosses. They are not leafy at all but rather flat and ribbon-like or heart-shaped and bilaterally symmetrical. The body is sometimes referred to as a thallus, a body without roots, stems, and leaves. Currently, body is much more commonly used than thallus. Bodies of thallose liverworts are stratified and tend to be much thicker than those of leafy liverworts and mosses. The side next to the substrate bears unicellular rhizoids, and many cells contain large oil drops. Cells in the side away from the substrate have no oil but are rich in chlorophyll. The cells are loosely arranged as an parenchyma with large air chambers that open to the exterior by means of large air pores. Air pores are not stomata, they have no guard cells and cannot be closed. Some species are even simpler. Spherocarpos texanus is a small, thin ribbon a few cells thick at the center, but the rest of the body is only one cell thick. It has simple rhizoids on the bottom but no internal air chambers and no scales or other types of vegetative differentiation. These plants are basically as simple as the sea lettuce alga, ulva, or the charophyte, coleochaete. The liverworts Palavicinia and Pellia are also this simple. Liverwort gametophores may be either bisexual, producing both Antheridia and Archegonia, or unisexual, depending on the species. Leafy liverworts may bear their gametangia either mixed with regular leaves or positioned on specialized side branches and surrounded by modified leaves. In thallose species, Antheridia and Archegonia may be grouped together and surrounded by a tube of chlorophyllous cells. Marchantia is probably the most familiar thallose liverwort because it grows easily on moist soil in greenhouses. Its gametangial production is particularly elaborate. Male gametophores of Marchantia produce an umbrella-shaped outgrowth called an antheridiophore. It has a stalk several millimeters tall, and dozens of Antheridia grow from its upper surface, each surrounded by a rim of sterile cells. Archegoniophores also are stalked, but their apex is a set of radiating fingers that project outward and droop downward. The underside has numerous archegonia. Archegoniophores and antheridiophores occur on separate plants in Marchantia. Antheridia and Archegonia are not produced by the same gametophyte. If sperm cells are carried to the archegoniophore by raindrop splashing, they swim into the archegonium neck and fertilize the egg. The zygote is retained and grows into a small sporophyte. Surrounding gametophore tissue expands with it temporarily, forming a protective sheath, and the archegonial neck expands into a calyptra. In Marchantia, sporophytes are so small that each archegoniophore can support many of them. The sporophyte generation. Little variability exists in the sporophytes of most liverworts, and their basic morphology is like that of mosses. In fundamental structure, however, the two groups are quite distinct. Most liverwort sporophytes have a foot, seta, and calyptra-covered sporangium, but the seta is extremely delicate, composed of clear, thin-walled cells that collapse quickly. Many members of Marchantiales have no seta at all. The sporangium is globose and while young is bright green and chlorophyllous, but it has no stomata. The outer layer of sterile cells is much thinner than in moss sporangia, often only one cell thick. The liverwort sporangium lacks a columella, the central mass of sterile cells found in mosses. Dehiscence occurs by means of 4 longitudinal slits, not by an operculum, and there are no peristome teeth. Within the sporangium, some cells do not undergo meiosis but rather differentiate into elaters, single, elongate cells with spring-shaped walls. When the sporangium opens, the elaters uncoil, pushing the spores out. All liverworts are homosporous. Strikingly simple sporophytes are produced in Riccia and Rixiocarpus. The zygote grows into a spherical mass within the archegonium of the gametophore body. No foot or seta is formed. Instead, the inner cells of this mass undergo meiosis and produce spores. As the surrounding gametophore and sporophyte tissues age, die, and decay, the spores are finally liberated. Figure 2023. Although gametophytes of thallose liverworts may become very thick, they are never solid. Rather, they have numerous chambers, and both carbon dioxide and water vapor can diffuse through them easily. Figure 2024. These are antheridiophores of Marchantia. As their name indicates, these structures bear antheridia. These are gametophytic tissues, and the antheridia are hidden within the upper surface. Low magnification view of antheridiophore with antheridia. Like antheridia in mosses and all other embryophytes, those in liverworts consist of a jacket of sterile cells surrounding fertile cells that differentiate into sperms. Figure 2025 archegoniophores of Marchantia are easily recognizable because they have finger-like segments radiating from the stalk. Archegonia are located on the underside. Each archegonium consists of a long, tubular neck and a slightly swollen base, all only one cell thick. Each archegonium contains only one egg. Figure 2026 liverwort sporophytes consist of foot, seta, and capsule, but no elaborate set of teeth as in mosses. Instead, the apex breaks into several segments, all of which curl back and release the spores. Division Anthoceratophyta, hornworts. Hornworts are a group of small, inconspicuous thalloid plants that grow on moist soil, hidden by grasses and other herbs. They rarely inhabit tree trunks or bare rock, therefore, one does not often encounter them unless looking for them specifically. Roadside cuts and stable eroded soil in shade are good sites for hornworts. Hornworts number approximately 100 to 150 named species in 5 or 6 genera, with Anthoceros and Phaeoceros being common examples. Many species names are probably synonyms, that is, several names applied to the same species because many hornworts have such variable growth forms that individuals of the same species can differ greatly under varied conditions. Figure 2027 spores and elaters of the liverwort Haplometrium hookeri. Figure 2028 The horns of a hornwort are sporophytes that grow continuously from a basal meristem. The lower part of the sporophyte is surrounded by gametophyte tissue. Hornworts superficially resemble thalloid liverworts, the gametophores of many species being ribbon-shaped and thin, without a distinct stem or leaves. However, hornworts never contain oil bodies, whereas liverworts almost always do. As in all other embryophytes, an alternation of heteromorphic generations occurs, with the sporophyte depending on the larger, photosynthetically active gametophyte, but hornworts are quite distinct from all other embryophytes, including the other non-vascular plants. One of the most striking features is the presence of a single large chloroplast in each cell as opposed to the numerous small plastids present in all other non-algal plants. The single chloroplast per cell is characteristic of many algae and is surprising in hornworts. Furthermore, hornwort chloroplasts have a pyrenoid, an algal feature absent in all other embryophytes. Hornwort chloroplasts probably represent the condition of the ancestors of all embryophytes. Unfortunately, we do not know what the chloroplasts of the earliest plants were like because they are not preserved in any known fossil. Table 26 Classification of Division Anthoceratophyta. Figure 2029 Hornwort cells are unique among embryophytes, each has just one plastid. Outside of hornworts, this feature is known only in algae. Although it may seem trivial, it must represent a significantly distinct cell-plastid relationship. The plastid's reproduction must be carefully coordinated with that of the cell, and a special mechanism is necessary to ensure that both progeny cells receive one each. Cells of privet leaf with many chloroplasts each. The gametophyte generation. As few as 3 or 4 protonema cells are produced before the gametophore phase is established in most species. Gametophores are always thin, at least along the edges. Only in the center do they become more than 4 or 5 cells thick. They may be shaped like a ribbon or a heart, or they may grow outward irregularly, forming a disc. The upper surface in many is smooth, but in others thin. Chlorophyllous lamellae grow upward. The gametophyte is parenchymatous, rather succulent but brittle. It does not tolerate drying. Gametophores of hornworts typically live for less than 1 year in temperate climates. They act as winter annuals, appearing in the cool, moist autumn months, growing during winter, producing sporophytes in the spring, and dying before summer. In some species, they form oil-rich tubers. As inner cells fill with oil and outer cells die, one thallus can produce several tubers. Internally, hornwort gametophytes have numerous chambers. Young plants have mucilage chambers formed as cells break down and their contents are altered chemically into mucilage. These become invaded by nostoc cyanobacteria. All hornworts form this symbiosis and presumably benefit by receiving nitrogen compounds from the nostoc. Gametangium development in hornworts is distinctive. Unlike all other embryophytes, the antheridial initials are not surface cells on the hornwort gametophyte. Instead, a special mucilage chamber forms near the upper surface, then cells lining the chamber grow into it and become antheridia. The chamber expands as the antheridia grow, and finally, its roof breaks. Only then are antheridia exposed to the environment. As sperm cells mature, sterile outer cells of antheridia transform their chloroplasts into chromoplasts, becoming orange or yellow. Archegonia are formed from superficial cells, but archegonia do not completely surround the egg as do the flask-shaped archegonia of other embryophytes. Rather, the egg lies below a short neck and neck canal but is surrounded by vegetative thallus cells. Even the neck cells are not particularly distinct but rather are difficult to distinguish from ordinary thallus parenchyma cells. Figure 2030. And in mosses, the initial cell in the formation of either an antheridium or an archegonium lies on the surface of the gametophyte. Then cell divisions change this single cell into a mass that has sterile jacket cells and reproductive central cells. This pattern occurs in all mosses, liverworts, and vascular plants. The gametophytes of flowering plants are so reduced that the pattern is not obvious. And the gametangia of hornworts are unique. The antheridia do not form on the true surface of the gametophyte, and the egg is not surrounded by discrete archegonial cells. After fertilization occurs, the zygote divides longitudinally. In mosses and liverworts, it divides transversely. The sporophyte generation. Similarities between sporophytes of hornworts and those of mosses or liverworts are not easy to find. Hornworts have a foot embedded in gametophore tissue, but there is no seta or discrete sporangium. Instead, just above the foot is a meristem that continuously produces new sporangium tissues. As the newly formed cells are pushed upward, they grow, differentiate, mature, and die. They are simultaneously being replaced by more cells from the basal meristem. Consequently, the sporangium is a long, horn-like cylinder, typically 1 or 2 centimeters long in Anthoceros and Phaeoceros, but up to 12 centimeters in some species. At the tip, the sporangium is mature and open as a result of dehiscence along 2 linear apertures. The outer layer of sterile cells is thick, up to 6 cells deep, and chlorophyllous. It has stomata in Anthoceros and Phaeoceros but lacks them in Notophilos, Dendroceros, and Megaceros. Spores are green, golden yellow, brown, or black, and in some species are multicellular when ready to be released. Hornworts have a columella as in mosses, but unlike the elaters of liverworts, those of hornworts are multicellular and do not have spirally thickened walls. The basal meristem is active over a long period, depending on moisture availability and temperature, and large numbers of spores can be produced by each sporophyte. Several attempts have been made to remove the sporophyte surgically from the gametophyte and grow it in laboratory conditions, but even though it is chlorophyllous, it dies. It is not known whether death is caused by a lack of minerals, insufficient photosynthesis, or absence of growth factors, vitamins, or hormones from the gametophyte. Figure 2031. The base of the sporophyte resembles a foot embedded in the gametophyte, and recently transfer cells have been discovered, therefore, active nutrient transport into the sporophyte must be occurring. Just above the foot is a meristematic region. At a higher level, equivalent to Figure 2032c, spores are mature. Figure 2032. A longitudinal section through the sporophyte. At the base is a meristematic region. Higher, above the basal meristem, sporocytes undergo meiosis. At higher levels the spores become mature and are then released. This type of continuous meiosis is unknown in plants other than hornworts. At the next level. 1. Gametophytes as complex plants. Every angiosperm has two very simple gametophytes, the male gametophyte and the female gametophyte. The gametophytes of bryophytes are much more complex, having leaves, stems, and rhizoids. Almost all plant biology is focused on sporophytes. But every plant without exception has a gametophyte stage. For independent study at the next level, I recommend reading about the gametophytes of every major group of plants, and the best place to start is in the following chapters of this book, plus Chapter 9 Flowers and Reproduction. After that, check online or in books on plant morphology. It is extremely difficult to study most gametophytes in person because, other than those of ferns, most gametophytes are small, live only briefly, and are rare at best. If you have a local expert that can help you find some in nature, that would be a great experience. 2. Bryophytes as wildflowers. I hope you pay some attention to the plants on campus or near your home. Perhaps you know the names of some trees and cultivated landscaping plants, maybe some wildflowers. People often notice mosses but have a difficult time learning their names. Many wildflower identification books list only 2 or 3 mosses and often with photographs in which important details cannot be seen clearly. But there are several excellent guides to bryophytes written for beginners. I recommend the following. 1. California Mosses by Bill Malcolm, Nancy Malcolm, Jim Shevick, Dan Norris. 2. A Trailside Guide to Mosses and Liverworts of the Cherokee National Forest by Paul Davison and Mark J. Pastrang. 3. Common Mosses of the Northeast and Appalachians by Carl B. McKnight, Joseph R. Rohrer, Kirsten McKnight Ward, and Warren J. Perdrizet. Even if you do not live in the areas specifically named by these guides, most mosses and liverworts are so widespread that these books are valid for many states. 3. Asexual reproduction. Bryophytes excel at asexual reproduction. In many species, almost any bit or piece of gametophyte tissue that happens to break off and then lands in a suitable site will grow into a new plant. In addition, many bryophytes produce small clusters of cells called gemmae that are easily dislodged by rain, wind, or animals and then grow into new plants that are clones of the original. Other species produce branches whose leaves fall off and grow, or the branches themselves fragment and grow. In all cases, gemmae or fragments are so tiny and lightweight that they can be blown hundreds of miles, allowing the plant to be dispersed over a wide area. In general, bryophyte species are much more widespread than seed plant species. Gemmae and asexual reproduction are significant alternatives to seeds and sexual reproduction. Summary. 1. True plants first originated at least 450 million years ago, having evolved from green algal ancestors. Necessary modifications would have included methods of water conservation, low surface-to-volume ratio, compact body, cuticle, and coordination of reproduction with presence of water. 2. The presence of plants on land changed the land as a habitat. With crowding, competition for light would have become important and an upright habit selectively advantageous. 3. The evolution of vascular tissue had profound consequences. Relatively strong, vertical stems could be supported. Large plant bodies could be integrated, and parts could differentiate and specialize. 4. Non-vascular plants are classified here in 3 divisions, emphasizing their differences and possible separate evolutionary origins: Bryophyta, Hepatophyta, and Anthoceratophyta. 5. All 3 groups of non-vascular plants are embryophytes. They have gametangia and sporangia in which only the internal cells differentiate into gametes or spores. A jacket of sterile cells is always present. 6. In all 3 groups of vascular plants, the life cycle consists of an alternation of heteromorphic generations. The gametophyte is the larger and more persistent and photosynthetically active phase. the sporophyte depends almost entirely on the gametophyte for carbon, energy, and minerals. 7. The seedy, stems, and leaf midribs of some mosses have cells that facilitate long-distance conduction. Leptoids transport sugars, and hydroids transport water. It is not known whether they are homologous or merely analogous to xylem and phloem. 8. Archegonia and Antheridia may be born on the same gametophore or on distinct, unisexual gametophores. 9. Neither the egg nor the zygote is released. The new sporophyte develops initially within the archegonium. When fully mature, it has a foot and sporangium. Many have a seta. 10. A moss spore grows into a filamentous protonema that produces buds that develop into thick parenchymatous gametophores with stems, leaves, and rhizoids. 11. Liverworts may be either thallose or leafy. Liverworts contain oil droplets, have bilobed leaves, and have no thick leaf midrib, no columella in the sporangium, and no operculum or peristome teeth. 12. Hornwort sporophytes differ from those of all other plants. They have a foot and a basal meristem, and thus, sporogenous tissue is formed continuously. 13. Hornwort Antheridia do not arise from surface cells, and the egg is not completely surrounded by distinct archegonial tissue. Mosses operculum peristome teeth protonema rhizoid seta thallus. Review questions. 1. Some vascular plants produce seeds, others do not. Vascular plants that do not produce seeds are known as, whereas vascular plants that do produce seeds are known as. Are there any plants that produce seeds but which do not have vascular tissue? 2. What are some of the modifications necessary if an alga is to become evolutionarily adapted to living on land? Is a single modification sufficient, or are several necessary? 3. Why would it be necessary for an evolutionary line to develop stomata and guard cells before it developed an extremely impervious cuticle? Why must vascular tissues precede the evolution of roots and active apical meristems? 4. The following organisms are often called mosses, but they are not actually closely related to mosses at all. What groups of plants do they actually belong to? A. Spanish moss. B. Club mosses. C. Slimy, bright green mosses, of ponds and slow-moving streams. D. Reindeer moss. 5. What are the three groups of non-vascular plants? How would you determine whether an unknown specimen is a vascular plant? 6. The non-vascular plants of this chapter are believed to be true plants, just as ferns, conifers, and flowering plants are true plants. However, there are two tissues that the non-vascular plants do not have. Which two tissues? 7. If the leptoids of mosses were found to contain a protein whose gene had the same nucleotide sequence as the gene that codes for P protein, would that be significant evidence for either the homology or analogy of leptoids and phloem? 8. You will see sporophytes only if you examine mosses closely. They look like green or brown, standing up on the green gametophyte, but sporophytes are. 9. Do mosses have an alternation of isomorphic or heteromorphic generations? That is, can you easily tell a moss gametophyte from a moss sporophyte? When we look at leafy green moss plants, what are we seeing, the gametophyte or the sporophyte? In a flowering plant species, would the equivalent stage be the plant or the pollen grains and megagametophytes? 10. The leafy, green moss plants that are so familiar are gametophytes, haploid plants. This is very different from flowering plants and other seed plants. Does a leafy green moss plant grow from a spore or from a fertilized egg? Does the moss plant have both a paternal parent and a maternal parent? 11. Draw a single moss plant, similar to the one in Figure 2010. Be certain to show the gametophyte and the sporophyte. Now draw one without the sporophyte, showing only the gametophyte. The sporophytes usually have only a very brief life, and after they shed their spores, the gametophytes let them die. 12. Draw and label the life cycle of a moss. Be certain to show gametangia and sporangia. Which parts are haploid and which are diploid? Where and when does meiosis occur? Plasmogamy? Karyogamy? 13. In the majority of mosses, which lack hydroids and leptoids, water is conducted along the of the plant by action. 14. The leafy, green moss plants, being gametophytes, have gametangia, structures that produce gametes. What is the name of the gametangium that produces sperm cells? The gametangium that produces egg cells? Can one single moss gametophyte bear both of these? Do some species have plants that produce only sperm cells? Other plants that produce only egg cells? 15. The sporophyte of a moss usually has a stalk called A and a simple apical sporangium called A. 16. Many people often think of mosses as plants adapted to rainy areas, areas that are usually wet. Are any mosses adapted to deserts? Can some mosses lose much of their water, the way a seed does before being planted, and still survive? 17. The liverwort Marchantia is one of the largest and most common. There is a good chance that you will find it if you search carefully in moist places. Is it a leafy liverwort or a thallose one? Describe its surface texture. If you are very lucky, you may find it with archegoniophores and antheridiophores. What are these structures, and how would you recognize them if you saw them? 18. Unlike Marchantia, some liverworts are as simple as a true plant can possibly get. Describe the body of Sphaerocarpos texanus. If you were shown one of these plants, how would you be certain it was not an alga? The point of this question is to have you think about how little difference there is between some algae and some true plants. 19. What are some of the ways in which liverworts differ from mosses? How do hornworts differ from both? Do the three have similar life cycles? 20. What are the horns of hornworts? What do they produce? They have a meristem. Where is it located? 21. An important consideration in the evolution of any organism is gene flow. What are some of the mechanisms by which genes move through the habitat in non-vascular plants? In a dense, cool forest, how strong are wind currents? Could they carry spores very far? What would you guess might be the maximum distance sperms can swim? How far can a raindrop splash a sperm or a spore? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 21. Vascular plants without seeds. Chapter opener image. This lower side of a leaf of bird's nest fern, Asplenium nidus, is covered with rows of sporangia. Within each sporangium, some cells undergo meiosis and produce haploid spores. Just as in bryophytes, these haploid spores are then released and must grow into gametophytes on their own. In seed plants, these spores would be retained, at least temporarily, on the leaf, and would start their development into gametophytes with nourishment from the leaf. In seed plants, megaspores would be retained until they develop fully into seeds. That never happens in the cryptogams. The leaf shown here will produce tens of thousands of spores that can be distributed great distances by wind. Outline. Concepts. Early vascular plants. Rhiniophytes. Zosterophyllophytes. The microphyll line of evolution, lycophytes. Morphology. Heterospory. Extant genera. The megaphyll line of evolution, euphylophytes. Trimerophytes. Origin of megaphylls. Manilophytes. Equisetophytes. Ferns. The term vascular cryptogams. Box 21-1 Botany and Beyond, Molecular Studies of the Evolution of Early Land Plants. Box 21-2 Botany and Beyond, Form Genera. Learning Objectives. After reading this chapter, students will be able to compare the interpolation hypothesis to the transformation theory. State the significance of rhiniophytes. List the characteristics that distinguish zosterophyllophytes from rhiniophytes. Name the 5 genera of lycophytes. Explain how trimerophytes differ from rhiniophytes. Summarize the telome theory. Describe the characteristics of ferns. Explain why the term vascular cryptogams may be used by some to refer to ferns and ferns' allies. Did you know? Most of the plants in this chapter are ferns. Ferns never produce flowers, seeds, fruit, or wood. Some garden ferns act like lilies. During seasons of stress, the foliage dies back and only the underground stems remain alive. They resprout when conditions improve. Ferns are an extremely diverse clade and occupy habitats as different as shady forests, the full sun of deserts, high mountains, and lakes. Concepts. What were the early vascular plants like? What features did they have, and what changes occurred as they diversified and evolved into the many types of plants alive today? Were they simple like some liverworts and hornworts and then gradually became more complex? Or were they completely different? With some giving rise to vascular plants by becoming more complex, whereas others gave rise to non-vascular plants by becoming simpler? Given all of the living plants present now plus the few fossils that we have from the time of the first embryophytes, what logical hypotheses can be proposed about the early embryophytes? First, all extant plants have a dibiontic life cycle. Each species has a multicellular gametophyte and also a multicellular sporophyte. Some algae, especially coleochaete, are monobiontic, having only one multicellular generation. A zygote undergoes only meiosis, producing more spores that will grow into new gametophytes. The zygote cannot undergo mitosis and cannot grow into a sporophyte. Thus, an important step in the evolution of embryophytes was the conversion of a monobiontic ancestor into dibiontic plants. A sporophyte generation had to come into existence. One hypothesis, the interpolation hypothesis, postulates that a small sporophyte came into existence when a zygote germinated mitotically instead of meiotically. The sporophyte generation would have gradually evolved in complexity while the gametophyte generation remained small. A sporophyte generation was inserted into the monobiontic life cycle. In this hypothesis, non-vascular plants such as some liverworts might be intermediates in the progression from green algae to vascular plants. For example, the liverworts Richia and Rixiocarpus have simple, almost algae-like gametophytes, and their sporophytes consist of just a small globosporangium with no foot or seta. The zygote undergoes several rounds of mitosis, then some cells undergo meiosis. It would take only a few evolutionary modifications to get such a simple sporophyte from an ancestor whose zygote merely underwent meiosis. If so, this sporophyte has been interpolated into the ancestor's monobiontic life cycle. From a Riccia-like condition, we could hypothesize that the sporophyte would evolve to be more elaborate, with a foot and seta, and then could later evolve to grow by an apical meristem, to branch, to live free of the gametophyte. It could be the ancestor of vascular plant sporophytes. This hypothesis requires that at least one of the non-vascular clades originated very early in the evolution of true plants. An alternative hypothesis, the transformation theory, postulates that after the dibiontic life cycle originated, both gametophyte and sporophyte became larger, more complex, and vascularized, in a life cycle with an alternation of isomorphic generations. No living plants have gametophytes that look like sporophytes, but many algae do, and some fossil plants did. Some fossils of early vascular plants bore gametangia and grew among other similar plants that bore sporangia. The transformation theory postulates that these early ancestors diverged into two clades, non-vascular plants in which sporophytes became much simpler and dependent on the gametophytes, and the rest of the vascular plants, in which sporophytes became increasingly elaborate whereas gametophytes became very reduced. Currently, one issue that is resolved is that all living and most fossil plants are dibiontic with an alternation of heteromorphic sporophytes and gametophytes. What we do not know is whether some liverworts or hornworts are relatively unchanged descendants of very early unvascularized ancestors or whether they originated later from more complex, vascularized ancestors and have since become simpler. This is discussed further after several early vascular plants have been described. Figure 21 the interpolation theory. The very earliest land plants were postulated as having no sporophyte. Instead, the zygote germinated by meiosis, producing haploid spores that grew into new gametophytes. At a later stage in evolution, the zygote would germinate mitotically and produce a simple sporophyte that in the early stages would have consisted of a sporangium and perhaps also a foot. This would have resembled a liverwort sporophyte. With continued evolution, the sporophyte would have become progressively more elaborate while the gametophytes became simpler. Figure 21-2 The transformation theory of the origin of the vascular plant life cycle postulates that in early land plants, gametophytes were upright and dichotomously branched, with epidermis, cuticle, and vascular tissue, just like sporophytes. With time, sporophytes became larger and more complex, and gametophytes became simpler. In the species illustrated here, gametophytes have become so small that the microgametophyte develops within the spore wall and the megagametophyte protrudes from the spore only slightly. With continued reduction, it is possible, but neither necessary nor inevitable, for the megaspore and its megagametophyte to be retained inside the megasporangium and remain on the parental sporophyte, an important step in the process of seed evolution. Early vascular plants. Rhiniophytes. The earliest fossils that definitely were vascular land plants belong to Cooksonia, a genus of extinct plants. These had upright stems that were simple, short cylinders, with no leaves. They had equal dichotomous branching, both branches being of equal size and vigor. Plants of Cooksonia had an epidermis with a cuticle, a cortex of parenchyma, and a simple bundle of xylem composed of tracheids with annular secondary walls. The ends of the branches were swollen and contained large, multicellular masses of sporogenous tissue surrounded by several layers of sterile cells. As in all plants, only the central cells were sporogenous, and the sporangium had to open to release the spores. Plants of Cooksonia were homosporous. There were no separate microspores and megaspores. Fossils that have these general characters are called rhiniophytes. Figure 21-3 A reconstruction of Cooksonia caledonica, the earliest known plant that had xylem, tracheids with annular secondary walls. Important features are its sporangia at the ends of branches, the lack of leaves, and the dichotomous branching. Rhinia and Aglaophyton were other rhiniophytes, early vascular plants similar to Cooksonia. They had a prostrate rhizome, upright naked stems, and terminal sporangia. Stomata and guard cells occurred in the epidermis, and there was a layer that appears to have been a cuticle. Figure 21-4 Branching patterns. If a stem forks, resulting in two equal stems, it is a dichotomy or dichotomous branching. It is pseudomonopodial branching if one stem is definitely larger and tends to form a trunk. If one stem dominates the system absolutely, it is monopodial branching, as in most seed plants. In the same rocks with Rhinia and Aglaophyton are fossils of similar plants. 2. Lionophyton and Cyathophyton were gametophytes, not sporophytes. The ends of their stems bore flattened cup-shaped areas that contained gametangia, both antheridia and archegonia, but not sporangia. These plants had upright, dichotomously branched stems, vascular tissue, stomata, and a cuticle. These were much larger and more complex than the gametophytes of any mosses, liverworts, or hornworts that are alive today. It is possible to suspect that because Rhynia-type sporophytes occur together with Cyathophyton-type gametophytes, they might have been alternate phases of the same species. If true, these plants had an alternation of isomorphic generations, and later evolution into seed plants involved reduction of the gametophyte to just a few cells and elaboration of the sporophyte into a more complex plant. These may have given rise to the non-vascular plants by reduction of both gametophytes and sporophytes. Another fossil from that same time, Horneophyton, had sporophytes with naked axes that branched dichotomously and were up to 20 centimeters tall. They had stomata and terminal sporangia. Each sporangium had a short columella, and just as in hornworts, the sporogenous tissue surrounded the columella on all sides and the top. The stem base was swollen, similar to a hornwort foot, except that it bore rhizoids. The similarities between the hornworts Anthoceros and Phaeoceros and Horneophyton raise the possibility that vascular plants may have arisen from hornwort-like ancestors. But it is also possible to think about these transitions in reverse, that hornworts evolved from species like Horneophyton by means of an elaboration of the gametophyte and a reduction of the sporophyte. Figure 21.5 Reconstruction of Aglaophyton major and Rhynia guineavoganii. They strongly resemble Cooksonia, and we know that they definitely had rhizomes, upright stems, and rhizoids. Inset shows the sporangium cut away, revealing spores. Reconstruction of the extinct plant Horneophyton and its terminal sporangia. At first, it was postulated that algae gave rise to true plants whose life cycle was dominated by the gametophyte and whose sporophyte was small and dependent, hemiparasitic on the gametophyte. It was postulated that this evolved into the vascular plants as the sporophyte became larger and more elaborate while the gametophyte became reduced. But it is possible that hornworts evolved by the reverse process, reading this series from right to left. Figure 21.6 Early vascular plants had two types of organization of xylem in their stems. In both, the center is a solid mass of xylem with no pith. This is a protostele. In an end-arch protostele, protoxylem is located in the center, metaxylem is on the periphery. End-arch protosteles occurred in the rhyniophytes. In an exarch protostele, metaxylem is located interior to the xylem mass and protoxylem is on the edges, as several groups. Exarch protosteles are found in fossils of zosterophyllophyte stems. Xylem structure of early vascular plants. Early vascular plants had two types of xylem organization. In both, the center is a solid mass of xylem with no pith, this is a protostele. In an end-arch protostele, protoxylem is located in the center and metaxylem differentiates on the outer edge of the xylem mass. Protoxylem is the xylem that differentiates while cells are small and narrow, and metaxylem differentiates after the cells have expanded for a few more hours or days and are larger. The other type of stele present in early vascular plants is an exarch protostele, with metaxylem located in the center of the xylem mass and protoxylem on the edges as several groups next to the phloem. Another type of stele, which did not evolve until later, is the siphonostele, one in which pith is present in the center, as occurs in the stems of ferns and seed plants. Because xylem is often preserved well in fossils, its characteristics of exarch endarch and protostele siphonostele are usually available for study. The xylem in many specimens of Rhynia and Aglaophyton is well preserved. It was a round cylinder without pith but with protoxylem in the center and metaxylem on the exterior, that is, an endarch protostele. All xylem cells were tracheids with annular thickenings. Around the xylem was a layer of phloem-like cells and then a parenchymatous cortex and epidermis. Zosterophyllophytes. Another group of early vascular plants are the zosterophyllophytes, named after the principal genus Zosterophyllum. They were small herbs without secondary growth. Many of their features were similar to those of rhyniophytes, but three characteristics make us think they were a distinct group. Their sporangia were lateral, not terminal, sporangia opened transversely along the top edge, and their xylem was an exarch protostele, that is, protoxylem on the outer margin and metaxylem in the center. Although these distinctions may seem minor, we are certain that in all the more recent plants with large leaves, sporangia are terminal rather than lateral. Thus, plants such as Rhynia may have been the transitions between algae and later seed plants, but Zosterophyllum was not. Instead, some of the simplest vascular plants alive today, the lycophytes, have lateral sporangia, and they may represent a line of evolution based on Zosterophyllum-like ancestors. Zosterophyllum plants grew as small bunches, only approximately 15 centimeters high. Upper portions of their stems had cuticle, ordinary epidermal cells, and stomata, but lower portions did not, presumably because they grew in swampy, marshy areas. Stems of Zosterophyllum were naked, branched dichotomously, and contained a small amount of xylem that consisted of tracheids with annular and scalariform secondary walls. Sporangial walls were several layers thick, and all spores were the same size, therefore, they must have been homosporous. Figure 21-7 Zosterophyllum rhianum plants were quite similar to those of rhiniophytes, but the ends of fertile branches bore numerous lateral sporangia, not a single terminal one. Also, sporangia opened by a suture that passed over the top of the sporangium, not up its side. Figure 21-8 proposed phylogeny of vascular plants without seeds. Table 21-1 Characteristics of rhiniophytes and Zosterophillophytes. Figure 21-9 This reconstruction of the Zosterophillophyte Rhybukia ovata shows several significant advances in body construction. The stems that bore sporangia were specialized, keeping sporangia close together and elevated into the wind. Other stems branched repeatedly, forming a large photosynthetic surface. Some branches were larger than others, and thus, branching was no longer strictly equal dichotomies. Other genera of Zosterophillophytes show that significant morphological changes evolved quickly. In Rhybukia, sporangia occurred together on the ends of specialized branches. In Chromaticollis, some branching was pseudomonopodial, larger, trunk-like shoots bore smaller, shorter lateral shoots. These are the morphological bases for producing cones and trunks. Several Zosterophillophytes had a smooth surface, but others had outgrowths called innations that ranged from quite small to long, thin scales. Innations increased the photosynthetic surface area of the plants, and in Asteroxylon, they contained stomata and a small trace of vascular tissue that ran from the stele through the cortex to the base of the innation. Figure 21-10 Reconstruction of Asteroxylon, an early lycophyte, showing its surface covered with innations, small flaps of photosynthetic tissue. In species without innations, stems were round and oriented vertically, not very good for harvesting sunlight. The microphyll line of evolution, lycophytes. Lycophytes represent a distinct line of evolution out of the early land plants that resembled Zosterophillophytes. Lycophytes have lateral sporangia and exarch protosteles, and thus, they may have come from a Zosterophyllum type of ancestor. Morphology. The earliest lycophytes were members of the genera Drepanophycus and Baragwanathia. They were similar to their presumed ancestors, the Zosterophillophytes, with an important difference: their innations were large, up to 4 centimeters long, and they contained a single well-developed trace of vascular tissue. Such innations must have increased photosynthesis, and they could be called leaves. However, leaf is an ambiguous term, and innations in the division Lycophyta are called microphylls for clarity. Micro- refers to their evolution from small innations, not to their actual size. In some plants, they were up to 78 centimeters long. This is not the line of evolution that led to ferns and seed plants. Microphylls are not the same as the leaves you are familiar with. Table 21-2 Classification of division Lycophyta. All species in this group are extinct. Another important advance was the evolution of true roots that allowed lycophyte sporophytes to anchor firmly, absorb efficiently, and thus to grow to tremendous size. Many extinct lycophytes such as Lepidodendron, Sigillaria, and Stigmaria had a vascular cambium and secondary growth. Their wood looked remarkably like the secondary xylem of pines and other living conifers, having a pith, rays, and elongate tracheids. However, the vascular cambium had one major flaw: its cells apparently could not undergo radiolongitudinal division, therefore, new fusiform initials could not be produced. As the wood grew to a larger circumference, cambial cells became increasingly wider tangentially. No fossil has ever been found with wood more than about 10 centimeters thick. After that much secondary growth, the cambial cells may have stretched so much that they could no longer function. Heterospory. In many extinct and extant lycophyte sporangia are clustered together in compact groups called cones or strobili, which protect them. Although many species remained homosporous, others became heterosporous, having microspores and megaspores that germinated to give rise to distinct microgametophytes and megagametophytes, respectively. Heterospory is a necessary precondition for the evolution of seeds. In the lycophytes Lepidostrobus and Lepidophlorus, the megaspore developed into a megagametophyte without enlarging. The megagametophyte existed completely within the wall of the original megaspore, which was up to 10 mm long. Furthermore, the megaspore in some species was retained within the sporophyll, protected by thick-walled cells of the sporangium. This is remarkably similar to ovules and seeds in seed plants, the most important difference being that in these lycophytes, the sporangium dehisced, and the megaspore wall cracked, exposing the archegonia. Sperm cells could swim to the egg during fertilization. Pollen tubes were unnecessary. Figure 2111 Drepanophycus was an early lycophyte that was still small and simple. Microfilson lycophytes are believed to have evolved as enations. Originally they were small, simple flaps of photosynthetic tissue. Later they became larger and were vascularized. See Figure 2120 for evolution of megaphils. Lycophytes are remarkable in that they represent an ancient line of evolution distinct from seed plants but having convergent evolution in several characters: leaves, roots, secondary growth, and almost seeds. In the Devonian and Carboniferous periods, this group dominated the swampy areas of Earth with extensive forests of large trees, but most became extinct. Currently, the entire division contains only 5 genera: Lycopodium, Phyloglossum, Selaginella, Isoetes, and Stylites. Figure 2112 During the Carboniferous period, Earth's vegetation was dominated by extensive forests of large lycophyte trees. None became truly massive like our redwoods, oaks, and elms, but the forests were both dense and vertically stratified. Flying insects were present, and early reptiles were just appearing. Amphibians were the dominant land animals. Figure 2113 Reconstruction of Lepidodendron showing a large tree with one distinct trunk and a well-developed root system. Leaves were sophisticated, and sporangia occurred in discrete strobili. Sigillaria was a large lycophyte tree in which the leaves usually were 1 m or more long. On the branch to the left, leaves have been left undrawn to reveal cones of sporangia. Extant genera. Lycopodium is fairly common in forests from tropical regions to the Arctic. All living species, approximately 200, are small herbs with prostrate rhizomes that have true roots and short upright branches. Microphylls are spirally arranged on their stems, and secondary growth never occurs. Sporangia may be arranged in cones or distributed along the shoots. All Lycopodium species are homosporous, a plesiomorphic trait. Spores germinate and grow into bisexual gametophytes that produce both antheridia and archegonia. In some species, gametophytes are green and photosynthetic; in others, they are subterranean and heterotrophic, nourished by fungi. Selaginella is less common in temperate North America, and its plants are smaller and easily overlooked or mistaken to be mosses. Probably the best-known species is the resurrection plant, S. Lepidophylla, which curls up, turns brown, and appears dead upon drying but uncurls and regreens when moistened. Unlike Lycopodium, Selaginella has the apomorphic condition of being heterosporous, and the megagametophyte develops inside the megaspore wall. The megaspore is not retained on the sporophyte, however, and is not seed-like. Microgametophytes also develop within the spore wall and consist of a single vegetative cell and an antheridium. Many flagellate sperms are produced and then released as the spore wall ruptures. Figure 2114 Wood of Sigillaria consisted of large tracheids and superficially resembled the wood of modern conifers. However, it was not as derived in its pitting and other features of its secondary